All right, hey guys. Today we are going to talk about each of the senses of the body. So we'll be going over the general senses of the body, how those are perceived, and then we're going to talk about how the special senses of the body operate. Okay. So first up, here's how we can kind of divide our senses in our body. So we have two general categories of general senses versus special senses. So with general senses, we have receptors that are distributed over a large part of the body, versus with special senses, we have specialized receptors that are located within specific organs as opposed to being generalized all over the body. With general senses, we have two major categories. We have somatic sensory mechanisms and visceral sensory mechanisms. The somatic ones are typically located within the skin, muscles, and joints, and those fall under 5 different categories of touch, pressure, proprioception, temperature, and pain. Our visceral fall into 2 major categories of pain and pressure. With special senses, we have our 5 major special senses of smell, taste, sight, hearing, and balance. So before we get into the receptors of the skin, let's talk about some of the different kinds of receptors that the body has. We have things called mechanoreceptors that respond to mechanical stimuli, such as the bending or stretching of receptors. We have chemoreceptors, which respond to chemicals. For example, odor molecules, which bind to chemoreceptors, allow us to perceive different kinds of smells. We have photoreceptors, which respond to light. We have thermoreceptors, which respond to temperature changes. And we have nociceptors, which respond to stimuli that result in the sensation of pain. So the general senses have sensory receptors that are widely distributed throughout the body. The general senses include the senses of touch, pressure, pain, temperature, vibration, itch, and proprioception, which is, as we mentioned before, the sense of movement and position of the body and limbs. Many of the receptors for general senses are associated with the skin, as we'll see here. Others are associated with deeper structures such as tendons, ligaments, and muscles. Okay. The simplest and most common type of sensory receptors that we have are these things called free nerve endings, which we can see right up here in some of the superficial layers of I'm sorry, not the superficial, but the deeper layers of the epidermis here. So we have free nerve endings which respond to painful stimuli, temperature, itch, or movement. So we have a couple different kinds of receptors like thermoreceptors and nociceptors up here. We have Merkel discs that are present at some of the ends of these free nerve endings, which detect light touch and some very superficial pressure sensations. We have hair follicle receptors that are present down here, which detect very, very light touch. So if something grazes or brushes against our hair, the hair follicle receptors are the ones that pick that up. And then we've talked about these before as well. We have several different kinds of specialized corpuscles or nerve endings that respond to different kinds of touch. So Meissner's corpuscles are involved in fine discriminative touch. We have Ruffini corpuscles, which we haven't mentioned before, but these detect continuous touch or pressure. And then finally, we have our deepest ones, Pacinian corpuscles, which detect deep pressure, vibration, and position. So pain is an interesting sensation. It can be categorized in a few different ways. It's categorized by a group of unpleasant perceptual and emotional experiences. There's typically 2 types. We have the first one, which is localized sharp pricking or cutting pain resulting from rapidly conducted action potentials. And then we have diffuse burning or aching pain, which results from action potentials that are propagated a little bit more slowly. Superficial pain sensations in the skin are highly localized as a result of the simultaneous stimulation of pain receptors and tactile receptors. Deeper visceral pain sensations are not highly localized because of the absence of tactile receptors in the deeper structures. Visceral pain stimuli are normally perceived as diffuse pain for this reason. Perception of pain can be distributed through anesthesia. Local anesthesia suppresses action potentials from pain receptors in local areas of the body through the injection of chemical anesthetics near a sensory receptor or nerve. Pain sensations can also be suppressed if loss of consciousness is produced. This is usually accomplished by general anesthesia, a treatment where chemical anesthetics that affect the reticular activity system are administered. Pain sensations can also be influenced by inherent control systems. Sensory axons from tactile receptors in the skin have collateral branches that synapse with neurons in the posterior horn of the spinal cord. Those neurons in turn synapse with and inhibit neurons that give rise to the spinothalamic tract, sensory pathway that relays sensations in the brain. For example, rubbing the skin in an area of injury stimulates the tactile receptors, which send action potentials along the sensory axons of the spinal cord. According to the gate control theory, these action potentials close the gate and inhibit action potentials carried to the brain by the spinothalamic tract. So in reality, rubbing the site of a pain response can actually help with introducing some kind of organic local anesthetic, if you will. The gate control theory may explain the physiological basis for several techniques that have been used to reduce the intensity of pain. Action potentials carried by the spinothalamic tract can be inhibited by action potentials carried by the descending neurons of the dorsal column system. These neurons are stimulated by mental or physical activity, especially involving movement of the limbs. The descending neurons synapse with and inhibit neurons in the posterior horn that give rise to the spinothalamic tract. Vigorous mental or physical activity increases the rate of action potentials in neurons of the dorsal column and can reduce the sensation of pain. Exercise programs are important components in the clinical management of chronic pain. Acupuncture and acupressure procedures may also decrease the sensation of pain by stimulating descending dorsal column neurons, which inhibit action potentials in the spinothalamic tract. The gate control theory also explains why the intensity of pain is decreased by diverting a person's attention, because then you cannot focus on those pain signals. So now let's talk a little bit about what referred pain is and where different areas of our body can actually tell us about sensations of pain that are occurring in deeper parts of our viscera. So referred pain is perceived to originate in a region of the body that is not the source of the pain stimulus. Most commonly, we sense referred pain when deeper structures such as internal organs are damaged or inflamed. This occurs because sensory neurons from the superficial area to which the pain is referred and the neurons from the deeper visceral area where the pain stimulation originates converge onto the same ascending neurons in the spinal cord. The brain cannot distinguish between the two sources of pain stimuli, and the painful sensation is referred to the most superficial structures innervated, such as the skin. We can link this back to our dermatological map as well. Lots of the segmental neurons that innervate different regions of the guts, for example, have their links with superficial structures like the skin that are associated with each of those spinal segments. A good example of this is somebody who has ulcerative colitis or Crohn's disease, who's experiencing a blockage in some part of their intestines, might experience back spasming or a back pain that's linked to a blockage that's occurring within their viscera. It's actually happened to my brother-in-law. He has Crohn's disease, and he had mentioned that he was having some really bad back spasms that he wasn't sure what they were linked to, but he felt that since he was in the hospital for prolonged periods of time, treating the initial condition, he thought he'd kind of just chalked it up to it being, part of him just being in bed for a few days. When in reality, that back spasm was actually linked to an inflammatory localized pain response or an area of referred pain that was going on with the blockage in his small intestine. And so he ended up having to get that part of his intestines removed through an intestinal resection. So areas of referred pain can have a lot of different applications. Referred pain is clinically useful in diagnosing the actual cause of the pain stimulus. So, for example, during heart attacks, pain receptors in the heart stimulated when blood flow is blocked to some of the heart muscle. Heart attack victims, however, often may not feel the pain in the heart, but instead perceive the cutaneous pain radiating from the left shoulder down the arm due to those pain receptors being localized near that area of the body, right? So we can see that area being associated with this orange region right here, okay? Then we have some other interesting areas of localized pain as well. So we have esophageal pain that can be linked to this upper left portion of the chest cavity, lungs and diaphragm kind of in the upper neck region, liver and gallbladder in this right neck region over here, okay? Liver and gallbladder just here, stomach just here, colon right in the middle around the navel area or umbilical region, appendix just here, and then we have this large orange area associated with our kidneys. The kidneys are kind of interesting too because they have a lot of their links to genitalia formation. So it makes sense where we would have a lot of area, a lot of surface area that kind of leads down into the genital region that can, link to either kidney or urinary bladder issues. Okay, So now let's talk about one of our first kinds of special senses, the olfactory epithelia. What I'll end up doing at the end of lecture today is I will kind of pause the recording here, and I'll get out the Atlas application. We'll go through some of these structures in close-up 3 dimensions so you guys can have this as part of your study guide. Olfactory, the olfactory bulb is an extension of the first cranial nerve, the cranial nerve number 1, the olfactory nerve, that extends into the frontal, not the frontal lobe, but it extends underneath the frontal lobe or inferior to that and lands on a structure called the ethmoid bone. The olfactory bulb has a bunch of nerve extensions that enter the foramina of the ethmoid bone called the cribriform plate. And those epithelia, those neurons will then extend down into the nasal cavity and have their nerve endings resting within the epithelia of the nasal cavity. Okay, so that's what we're seeing here. Typically, that occurs mostly in the superior nasal conchae, right here. Right. So then upon closer inspection, we can see that the olfactory bulb just carries multiple pathways of neurons. So we have multiple extensions of unipolar neurons that have the soma of the neural body embedded within the olfactory epithelia, where we have various kinds of cilial cells that are embedded within a specialized kind of mucus that will pick up different kinds of odorants or smell molecules that dissolve within the tissue of the epithelia. And depending on the nature of that smell, whether or not it can dissolve in water or dissolve in lipids, that will have different effects on how the brain perceives those things. So the olfactory epithelium here, we've got the little extensions of the olfactory neurons here that have their roots within the mucosal layer of that epithelia. And then our tract pathway comes back up here and synapses onto the olfactory bulb, just there, passing through the cribriform plate of the ethmoid bone. Okay, so now let's talk about taste. All right, so taste is kind of interesting. Multiple nervous pathways are associated with taste sensation. We can typically divide up the tongue into an anterior 2/3 and then a posterior 1/3. So the sensory structures that detect taste stimuli are the taste buds. The taste buds are oval structures located on the surface of certain papillae, which are enlargements on the surface of the tongue. Okay, so the little taste buds are these little guys right here that kind of emerge out on different parts of the papillae. So they occur all over the place. Taste buds are also distributed throughout other areas of the mouth and pharynx, such as on the palate, the root of the tongue, and the epiglottis. Each taste bud consists of 2 types of cells. Specialized epithelial cells form the exterior supporting capsule of each taste bud, and the interior consists of about 40 taste cells. Each taste cell contains a hair-like process called taste hairs that extend through a tiny opening in the surrounding stratified epithelia called a taste pore. Dissolved molecules or ions bind to receptors on the taste hairs and initiate action potentials, which sensory neurons then carry to the insula of the cerebral cortex. Taste sensations are typically divided into 5 basic types. We have sour, salty, bitter, sweet, and savory or umami. Although all taste buds are able to detect all 5 of the basic taste sensations, each taste bud is usually most sensitive to one class of taste stimuli. Presumably, our ability to perceive many different tastes is achieved through various combinations of these 5 basic types. Heat damage to the tongue epithelial tissue can cause injury or even death to epithelial cells, including taste cells and the taste buds. If the epithelial cells are only damaged, taste sensation usually returns within a few hours to a few days. If the cells die, it takes about 2 weeks for the epithelial cells to be replaced. Many taste sensations are strongly influenced by olfactory sensations. This influence can be demonstrated by comparing the taste of some food before and after pinching your nose. It's easy to detect the taste, or to detect that the sense of taste is reduced while the nose is pinched. I don't know about you guys, but anytime I had to drink prune juice when I was a kid, that was what I always did, is I always pinched my nose. So we've got each of our little taste hairs right here within our individual taste cell, okay? And so there's different regions of the tongue that will pick up different kinds of taste too. So as I mentioned before. And here we've got a representation of the different neural pathways for how different parts of taste are perceived. So with the tongue, kind of our anterior 2/3 right here is really sensed by the 5th and 7th cranial nerves, so the trigeminal nerve and the facial nerve just here. And then the posterior third is innervated via the hypoglossal nerve, okay? And then we have a little tiny portion back down here that's kind of leading into the epiglottis, which is innervated by the vagus nerve. I'm sorry, not hypoglossal, glossopharyngeal. Hypoglossal is cranial nerve number 12. Glossopharyngeal is number 9. All right, so now let's talk about sight, okay? So the eyeball is kind of an interesting structure, and this is, again, another cool one that we'll get to look at in 3 dimensions here in a bit. So from a superficial view, we'll look at our left-hand image and then go down here and then over to our large kind of sagittal view of the eye. So we've got our eyebrow and eyelashes, which really serve to keep debris out of the eye. Eyebrows are really good, or maybe they are, maybe they're not, depending on you know, how sweaty you are, but they're typically used for keeping things like sweat and other kinds of debris from running down the surface of the skin and getting into the eye. Same thing with eyelashes as well. Eyelashes are designed to keep little floating particles and debris out of your eyeballs. Unless you're like me, where every morning you wake up and you have an eyelash in your eye or something like that. I usually have an eyelash in my eye at some point. It's quite annoying. So we've got our eyelashes here. We have the center of our eye that can constrict and dilate to let different amounts of light into our eye, called the pupil. We have the iris surrounding the pupil right here. So the iris can take on different kinds of color based on genetic expression. We have this white portion of our eye called the sclera. And then we have the lateral and medial angles of the eye just here, and then with our eyelids lower and upper both. So in the medial angle of the eye, we have connections to the lacrimal canaliculi. So these are just fancy words for our tear ducts. We have the lacrimal sac right here, which is linked to those ducts as well. And the lacrimal gland, weirdly enough, sits on the upper lateral aspect of our eye at an oblique angle. So we have vasculature that links to the lacrimal canaliculi behind the actual eye itself. So it goes behind the sclera. So those lacrimal ducts will secrete tears, and we secrete tears without crying or anything like that. We use tears to help lubricate the eye as well. Those can actually be linked to the nasolacrimal duct, which can produce mucus in the nasal cavity or snot. So sometimes if you get something in your eye, or you inhale something through your nose that's like really spicy or something like that, you can sometimes produce tears, or you can produce a lot of snot as a result of that, or as a result of the opposite stimuli too. All right, so let's go through our little sagittal section of the eye right here. So we have our upper musculature, the orbicularis oculi muscle. So one of our sphincter muscles that can help us constrict our eyes a little bit. We've got our eyebrow just up here. And then we have some musculature that we'll go over here in just a minute. And we'll kind of spend most of our time talking about these other structures over here. So structures of the upper eyelid include the conjunctiva over the eye. So this is a set of connective tissue that surrounds the cornea and the anterior chamber. The conjunctiva of the eyelid just here. We have the sebaceous gland just here, which can produce various kinds of sebaceous secretions, various kinds of oily secretions. We have a connective tissue plate that's here as well, along with our cornea and then our eyelash. All right. With the lower eyelid, skin of the lower eyelid, loose connective tissue just here, orbicularis oculi just there, connective tissue plate along with the conjunctiva of the eyelid just here. So similar structures with the sebaceous glands and connective tissue just around there. Here we have some of the musculature of the eye. So as I mentioned before, we've already talked about this with our muscular lab. And we've seen a little bit of this in the app as well, but we'll get a better look at that in just a minute. But the musculature of the eye is actually fairly straightforward. We have lots of rectus muscles that exist in these kind of long rod-like configurations. And when those muscles contract, they just pull the eye in one direction. So the superior rectus, when that contracts, will cause the eye to rotate upwards, versus the inferior rectus, when that contracts, will cause the eye to tilt downwards. Then we have the lateral and then the medial rectus, which would be on the other side, which will move the eye in the lateral to right configurations. And then we're able to rotate the eye and use oblique angles by superior and inferior oblique contractions of that muscle, so the eye can kind of rotate and look down to the midline, the middle corner, or look up to the upper right midline of the corner, or up the upper right corner of the eye, sorry. Okay, So here, this figure talks about the various connective tissue or vascular sheaths of the eye. So we call these little sheaths tunics, all right? So we have three major tunics. We have the fibrous tunic, we have the vascular tunic, and then we have the nervous tunic. The nervous tunic's easy because only the retina or the optic nerve is going to be part of the retinas or part of the nervous tunic just here. The vascular tunic, which sits superficial to that, consists of the iris, the ciliary body, which we'll get to here in a minute, and then the choroid plexus. Choroid plexus carries a lot of the vasculature that helps nourish the eye and supply it with blood flow. The ciliary bodies here are going to be used to change the shape of the lens. And so that'll change how light enters the eye and how it can be focused onto the retina for visual acuity. We have the anterior chamber and the posterior chamber of the eye, which are filled with aqueous humor, or kind of like a thick, viscous liquid. The reason for that is so that we can change how light enters the eye so it doesn't diffract or shatter too widely, so we can focus all of that light into a specific point. And the better that we can do that, then the better our visual acuity is. If you're like me and you wear glasses, then your eyes don't really do that all that well. Sometimes you can have different issues that can change the shape of the cornea here. So this outer kind of thick layer that covers our anterior chamber. If you have a thickening or a misshapening of the cornea, that can result in something called an astigmatism. And astigmatisms will just change how light enters the eye, and it can change how the lens can try and focus that light into your retina. Then we've got our fibrous tunic, which is made of the sclera, the outer white connective tissue of the eye, and then the cornea, this little layer right here that covers the anterior chamber. Okay. And then we've got our pupil here that dilates and constricts to allow for light to enter and hit the lens. And then the lens can change shape based on the activity of the ciliary bodies to focus light onto a specific point in the eye. And so inside the eye, we have the vitreous chamber here filled with vitreous humor and lots of vasculature. Okay, so all of our vitreous humor is this stuff here. So this would be inside of the eye if it was actually in your skull. So here with the ciliary body, so basically the ciliary muscles and suspensory ligaments will serve to either compress the lens or allow the lens to relax. Typically, the thicker the lens, then that allows for light to be focused more closely, and the flatter the lens, the light can be focused a little bit more farther back into your eyeball. And we'll describe kind of what that means here in a sec. We have different ways of constricting the smooth muscle of the eye as well. So we have circular smooth muscles and radial smooth muscles that will constrict the iris or relax the iris. Okay, I'm sorry, the pupil. So when these, and so they're operated by different parts of the nervous system as well. The sympathetic nervous system contracts the circular smooth muscles, and the parasympathetic system contracts the radial smooth muscles to allow the pupil to dilate. So here we have the structure of the rods and the cones of the eye. So these are the parts of these are cells, I'm sorry, that respond to and interpret different kinds of color information within the eye. Okay. Let's see here. So the pigmented retina with the choroid keeps light from reflecting back into the eye. The sensory retina contains photoreceptor cells as well as numerous interneurons. Photoreceptor cells, which include rods and cones, respond to light. Over most of the retina, rods are 20 times more common than cones. Rods can function in dim light because they are very sensitive, meaning they require lower levels of light to be stimulated. Rods, however, do not provide color vision. Cones require much more light, and they do provide color vision. There are three types of cones, each sensitive to a different color: blue, green, or red. The interneurons of the sensory retina, some of which are identified in figure 9.12 of your book, regulate and relay changes in photoreceptors along the visual pathway. So a lot of science has gone into understanding which of these cells, either cones or rods, are responsive to different forms of light. So the color of dashboard lights in your car is actually designed with that in mind. The outer segments of rod and cone cells are modified by numerous foldings of the cell membrane to form these discs, right? So we can see these discs here, all right? Rod photoreceptors contain a photosensitive pigment called rhodopsin. which means purple pigment. Rhodopsin consists of a protein, opsin, loosely bound to a yellow pigment called retinol. When exposed to light, retinol changes shape, which then changes the activity of the entire rhodopsin molecule. This change in rhodopsin stimulates a response in the rods, resulting in vision. Retinol then completely detaches from opsin. Energy is required to reattach retinol to opsin and return rhodopsin to the form it was before it was stimulated by light. Okay. The manufacture of retinol in rods takes time and requires vitamin A. In bright light, much of the rhodopsin in rods is dissociated. Opsin and retinol are separated. For example, suppose you go into a dark building on a bright day. It will take several seconds for your eyes to adjust to the dark, and opsin and retinol reassociate to form rhodopsin in the rods, which can then react to the dim light. A person with a vitamin A deficiency may have a condition called night blindness, characterized by difficulty seeing in dim light. Night blindness can also result from retinal detachment, which is the separation of the sensory retina from the pigmented retina. Retinal detachment affects the periphery of the retina, where the rods are located, more than the center of the retina, where the cones are located. Because the rods are more sensitive to the cones than light, retinal detachment affects vision in low light to a greater extent than vision in bright light. The photosensitive pigments in cone cells are slightly different from those in rod cells. The pigments in cone cells are sensitive to colors. Each color results from stimulation by a certain wavelength of light based along the electromagnetic spectrum. Three major types of color-sensitive opsin exist, and they're sensitive to blue, red, or green. The many colors we can see result from the stimulation of combinations of these three types of cones. As stated earlier, the sensory retina also contains interneurons, including bipolar cells, horizontal cells, and ganglia. The rod and cone cell synapse of bipolar cells of the sensory retina. These and the horizontal cells of the retina modify the output of the rod and cone cells. For example, this modification helps us perceive the borders between objects of contrasting brightness. The bipolar and horizontal cells synapse with ganglia, whose axons converge at the posterior of the eye to form the optic nerve. So here we can see we have that process that I mentioned before with the dissociation of rhodopsin, of opsin, and retinol when they're exposed to light. And then when they are not exposed to light, then these can be reabsorbed or resumed back into their original shape of rhodopsin with the inclusion of ATP. Okay. When the posterior region of the retina is examined with an ophthalmoscope, two major features can be observed: the macula and the optic disc. Okay, so here, that's what we've got represented here. The macula is a small spot near the center of the posterior retina. In the center of the macula is a small pit, the fovea centralis. The fovea centralis is the part of the retina where light is most focused when the eye is looking directly at an object. The fovea centralis contains only cone cells, and the cells are more tightly packed than anywhere else in the retina. Hence, the fovea centralis is the region with the greatest ability to discriminate fine images, which explains why objects are best seen straight ahead. Okay. The optic disc is a white spot just medial to the macula, through which a number of blood vessels enter the eye and spread over the surface of the retina. This is also the spot at which axons from the retina meet, pass through the two outer tunics and exit the eye as the optic nerve. The optic disc contains no photoreceptor cells and does not respond to light. It is therefore called the blind spot. A small image produced or projected into the blind spot cannot be seen. And so the next figure that we'll see here, this is a way that you can kind of test the blind spot in your eyeball. So if you want, on your screen right now, you can close your right eye, hold the drawing in front of you or on your screen in front of your left eye, and stare at the plus symbol. Move your screen away from your eye. At a certain point, when the image of the spot is over the optic disc, the red spot will disappear. And that's how you can figure out where your blind spot is in your vision. Okay. So now let's talk a little bit about focal points. Okay. So when we focus on an object that's far away or distant, what ends up happening is the ciliary muscles in the ciliary body are relaxed. But counterintuitively, what that results in is a high tension in the suspensory ligaments. So what that ends up doing is causing the lens to be flattened. When the lens flattens, we shift the focal point of our light being crossed over here. So that's where each of these wavelengths of light cross, that's our focal point. We shift that back closer towards the fovea centralis. and that's going to allow us to focus on objects or images that are farther away. Conversely, if we are trying to focus on an image that's closer up, the ciliary muscles in the ciliary body contract, moving the ciliary body toward the lens. So the ciliary body moves towards the lens here. But as a result, the suspension or the tension in the suspensory ligaments becomes low. So that allows for the lens to relax in shape, becoming more oblong, and that shifts the focal point more anteriorly. So that allows for a larger object to be focused onto the fovea centralis at a greater degree of acuity. And here we have some images that you can test for color blindness. So if you're looking at this image, if you have normal vision, you should be able to see a 74 present here. But if you have red-green color blindness, what you might end up seeing is the number 21, okay? Versus this same test where we have normal vision, if you can see 5, if you have red-green color blindness, you'll typically see a 2 represented here. In this figure, we have a representation of the visual pathways in the brain, so how the brain perceives information from each of the eyes. So for the first part up here, each visual field is divided into temporal and nasal halves. We have our nasal halves right here, so the darker colors, and the temporal halves, which are the lighter colors. After passing through the lens, light from each half of a visual field projects to the opposite side of the retina, stimulating receptors. Boom, boom. Axons from the retina pass through the optic nerves to the optic chiasma, where some cross. Axons from the nasal retina cross, and those from the temporal retina do not. So the temporal retina end up staying here, and they end up crossing. Okay. Optic tracts extend from the optic chiasm, with or without crossing, to the thalamus. Collateral branches of the axons in the optic tract synapse in the superior colliculi of the midbrain. Optic radiations extend from the thalamus to the visual cortex of the occipital lobe. And then finally, the right part of each visual field, dark green and light blue, projects to the left side of the brain. And then the left part of each visual field, light green and dark blue, projects to the right side of the brain. So it's kind of interesting that both halves of our occipital lobe are going to be interpreting two halves of the information from our visual field. So our dark blue and light green get interpreted on the right side, and then dark green, light blue get interpreted on the left side. Kind of interesting. So here we have that same representation showcased. So we've got our optic nerves here, which have been severed, kind of close to the optic chiasm here. So we have our optic chiasm that passes back here to the occipital lobe, and the same thing that passes over here, okay? Where we have our optic radiation and then our visual cortex. And here we can see the thalamus present for synapsing those connections. Okay. So here we have representation of monocular vision versus binocular vision. So left monocular vision, right monocular vision, and then the field of range for binocular vision represented here. Okay, so now finally, let's get into one of our last special senses, which is a sensation of hearing, but then also by association, we have the special sense of balance associated with this too. So with our ear, we have our external auricle, which is composed of cartilage, and we have an auditory canal, which leads to the structure of our inner ear. Right? So we have our external ear, middle ear, and then inner ear. So sorry, middle ear, inner ear is just here. The middle ear is composed of a membrane and then 3 separate bones or ossicles that are linked together to form kind of a kick drum style association with this membrane. So a sound comes in and hits this tympanic membrane in rhythmic fashion that causes a connection with these 3, inner ear bones. We have the malleus, which is associated with the tympanic membrane, the incus, which is the middle bone, and then the stapes just here. Each of these bones will rotate around one another as sound hits against the tympanic membrane. But interestingly enough, the tympanic membrane isn't what we're actually hearing. What we're hearing and what we're interpreting as sound is the movement of these ossicles that push against fluid inside of the inner ear. This is where all of the real hearing happens. So we have these other features called semicircular canal. We have, the chordae tympani, which is the association or the ligamentous association between the malleus and the tympanic membrane. Then we have the vestibule, large opening chamber that's linked to the stapes just here. Then we have our cochlea, where sound can reverberate and move around in. And then we have our vestibulocochlear nerve, which is hooked up to our inner ear, which is how we interpret sound. Okay. We also, oh, sorry, I have this little note here about the auditory or eustachian tube. So the eustachian tube, if you've ever changed an elevation rapidly, what will sometimes happen is we build up pressure inside of our skull. So in order to equalize the pressure with our surroundings, what we end up doing is we can open or link or open the connection between our brain case and our mouth through, kind of popping your ears or opening your jaw really wide, which will cause the eustachian tube to open up and allow pressure to equalize from the inside of the skull with the outside environment. Okay, so here we have the sections of our cochlea. The cochlea is what's mostly associated with our sense of hearing, right? And then the vestibules are what we use to kind of coordinate our sense of balance. So we'll get to those structures here in a second. So really, we can summarize hearing as just just movement of sound through fluid. So inside of our cochlea here, we have these fluid-filled canals, right, that will end up shifting based on sound waves and can change the frequency at which they shift based on things like pitch or tempo within a sound frequency. That pitch or change will trigger movement in these little microvilli of these cochlear nerve endings here that are linked to the vestibulocochlear nerve. And depending on how these microvilli are stimulated, that will then trigger a corresponding response with the vestibulocochlear nerve, right? Starting up here in the left, we have our overall structure of our inner ear. So we have these large semicircular canals which are linked to the cochlea and the vestibule down here. So we have our oval window and our round window represented here, and then the apex or the end of our cochlea, right? We have our cochlear nerve, which comes down and innervates the cochlea, and then the vestibular nerve, or that part of the vestibulocochlear nerve that comes down and innervates the vestibular region, all right? So cross-section through our cochlea here, we have this membranous labyrinth structure. We have the periosteum of bone, inner lining of the bony labyrinth here. We have the scala vestibulae, which is filled with perilymph, so that's the fluid that's moving around in the inner ear. And then we have the vestibular membrane, just here. We also have a tectoral membrane and cochlear duct filled with endolymph. We have a spiral ligament, a basilar membrane, and the scala tympani, which is also filled with perilymph in the inferior aspect just here. And then we have our cochlear ganglia right here, which is associated with the various nerves that are linked to these spiral organs, which give us our sense of hearing. Okay, so we've got our cochlear duct right here, vestibular membrane, tectoral membranes, all the same structures. But here, the spiral organs, these are the guys that are going to be interpreting our sense of sound, right? So we've got these little microvilli structures. We have our hair cell right here, which is associated with them. And then we have various kinds of nerve endings and synapses that form with various cochlear nerve endings here. So since they're sensory neurons, all of the direction of the nervous signal is going to be coming away from the hair cell, just here. And we have supporting tissue and supporting cells that can help provide support to those tissues. So here, this is a good overview of how sound is going to end up working within our ears. So the first part is sound waves strike the tympanic membrane and cause it to vibrate, which causes a chain reaction here with the inner ossicles. Vibration of the tympanic membrane causes the malleus, the incus, and the stapes to vibrate. The base of the stapes vibrates in the oval window. Okay. Vibration of the base of the stapes causes the perilymph and the scala vestibuli to vibrate. Boom, 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 boom. Okay. Vibration of the perilymph causes the vestibular membrane to vibrate, which then causes vibrations in the endolymph. Boom, 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 boom. Okay. Vibration in the endolymph causes displacement of the basilar membrane. Short waves, or high-pitched frequencies, cause displacement of the basilar membrane near the oval window, and longer waves, which are low-pitched, cause the displacement of the basilar membrane some distance from the oval window. Movement of the basilar membrane is detected in the hair cells of the spiral organ, which are attached to the basilar membrane. So vibrations of the perilymph and the scala vestibuli and of the basilar membrane are transferred to the perilymph of the scala tympani. Vibrations in the perilymph of the scala tympani are transferred to the round window where they become dampened so that we don't completely blow out our inner ear. Okay, so here we have a representation of different sound pathways and how they're interpreted via the auditory cortex. So sensory axons from the cochlear ganglia terminate in the cochlear nucleus of the brainstem, just here. Okay, so cochlear nucleus of the brainstem. Axons from the neurons in the cochlear nucleus project to other brainstem nuclei or to the inferior colliculus. Okay, so boom, just up to here. Axons from the inferior colliculus project to the thalamus, which then go up here, so boom, boom. And then thalamic neurons project to the auditory cortex, which is in the temporal lobe, just here, where we interpret that sound. So deafness can come from a variety of different regions. You can have structural deafness, which can be linked to things like conductive deafness, or you can have sensory neural deafness. So hearing impairment can have many causes. In general, there's two categories of hearing impairment, so conduction deafness and sensory neural hearing loss. So conduction deafness results from mechanical deficiencies, for example, destruction of the ligament that holds the malleus and the incus together, versus sensory neural hearing loss, which is caused by deficiencies in the spiral organ or nerves. For example, loud sounds that can damage the delicate microvilli of the hair cells, leading to destruction of the spiral organ. So what's more common is a sensory neural deafness versus the structural leading to conductive deafness. And now finally we reach balance. Okay. So the sense of balance or equilibrium has two components, so static equilibrium and dynamic equilibrium. Static equilibrium is associated with the vestibule and is involved in evaluating the position of the head relative to gravity. Dynamic equilibrium is associated with the semicircular canals and is involved in evaluating changes in the direction and rate of head movements. So each part of our semicircular canals and the vestibule are going to be used for different kinds of balance. Okay, the vestibule of the inner ear can be divided into two chambers, the utricle and the saccule. Each chamber contains specialized patches of epithelium called the maculae, which are surrounded by endolymph. The maculae, like the spiral organ, contain hair cells. The tips of the microvilli of these cells are embedded in a gelatinous mass, often called the otolithic membrane. The membrane is weighted by otoliths, little ear stones, which are particles composed of protein and calcium carbonate. The weighted gelatinous mass moves in response to gravity, bending the hair cell microvilli and initiating action potentials in the associated neurons. The action potentials from these neurons are then carried by axons of the vestibular portion of that cranial nerve number 8, the vestibulocochlear nerve, where they're interpreted as a change in the position of the head. For example, when a person bends over, the maculae are displaced by gravity, and the resultant action potentials provide information to the brain concerning the position of the head. So that's what we end up seeing here. By tipping over, the position of those little otoliths here changes and moves against the macula here, causing those action potentials to shift because of the stimulation of these supporting cells and these hair cells down here. Okay, so we've got our vestibule just here, the utricle and the saccule just here. So we have our macula with hair cells and supporting cells just here. Then we have the gelatinous mass containing our otoliths. All right, so same kind of microvilli that we see in the cochlea. All right. The semicircular canals are involved in dynamic equilibrium, and there's 3 in each ear. The semicircular canals are placed at nearly right angles to one another, enabling a person to detect movements in essentially any direction. You can kind of think of like a gyroscope, for example. It's like that's what we're seeing here. The base of each semicircular canal is expanded into an ampulla. Within each ampulla, the epithelium is specialized to form a crista ampullaris. Each crista consists of a ridge of epithelium with a curved gelatinous mass, the cupula, suspended over the crest. The cupula is structurally and functionally very similar to the maculae, except that it contains no otoliths, unlike the other structures. Let's see. So the hair-like follicles, or I'm sorry, the hair-like microvilli of the crista hair cells are embedded in the cupula. The cupula functions as a float that is displaced by endolymph movement within the semicircular canals. As the head begins to move in one direction, the endolymph tends to remain stationary, while the cupula moves with the head. The difference displaces the cupula in a direction opposite that of the movement of the head. As movement continues, the fluid will then catch up. When the movement of the head and the cupula stop, the fluid continues to move due to momentum and inertia, displacing the cupula in the direction of the movement. Movement of the cupula causes the hair cell microvilli to bend, which initiates the depolarization of the hair cells. This depolarization initiates action potentials in the vestibular nerves, which then join the cochlear nerves to form the vestibulocochlear nerves. Continuous stimulation of the semicircular canals, as occurs, with things like motion sickness or with constant movement, can cause motion sickness, which can be characterized by things like nausea and weakness. The brain compares sensory input from the semicircular canals, eyes, and position receptors in the back of the head or in the back and lower limbs. Conflicting input from these sources will lead to motion sickness. Okay, so we got here, so we have, oh my gosh, who is this? She's a famous athlete. Let's see. Oh, it's not listed in the book. She's one of those, the famous gymnasts who competed for the US in the Olympics. Yeah. So what's interesting is that obviously all of this can kind of change with age. So we'll get to that in a little bit here. All right. So let's talk about some of these disorders. So we can have infections of places like the eye and the ear. So trachoma, it's a type of conjunctivitis caused by chlamydia, leading cause of infectious blindness in the world and transmitted by contact or flies. And you can have styes that develop, which is an infection of the eyelash hair follicle. So defects of focus, alignment, or color vision. You can have myopia, which is nearsightedness, ability to see close but not distant objects, caused when refractive power of cornea and lens is too great relative to the eye length. That's what I have. Hyperopia, farsightedness, ability to see distant but not close objects, caused when the cornea is too flat or the lens has too little refractive power relative to the length of the eye. And then finally, you can have an astigmatism, or cornea or lens that's not uniformly curved, so the image can't be sharply focused. And then strabismus, one or both eyes are misdirected, which can result from weak eye muscles. And the best way to correct that is to cover up the stronger eye with an eye patch and force the weaker eye to compensate for that. And we can have various forms of blindness. So cataracts, clouding of the lens as a result of advancing age, infection, or trauma, most common cause of blindness in the world. You can have macular degeneration, which is a loss of sharp central vision. Peripheral vision is typically maintained. Leading cause of legal blindness in older Americans, most causes not known. We do know that there is a link to eye color. So typically people with lighter irises are more prone to developing macular degeneration. You can have diabetic retinopathy, involves optic nerve degeneration, cataracts, retinal detachment, often caused by blood vessel degeneration and hemorrhaging within the sclera. You can have retinal detachment, separation of sensory retina from pigmented retina, relatively common problem, which may result in vision loss. And then you can have hearing or ear disorders. You have tinnitus, phantom sound sensations such as ringing in ears, which is a common cause of constantly listening to loud sounds or being exposed to loud sounds. And then Meniere's disease, vertigo, hearing loss, tinnitus, and a feeling of fullness in the affected ear. Most common disease involving dizziness from the inner ear. Most are unknown, but may involve fluid abnormalities within the ears. So let's talk a little bit about what happens to all this stuff with age. So as we get older, both the general and special sensory functions gradually decline. Among the general senses, free nerve endings and hair follicle receptors in the skin remain largely unchanged. However, the number of Meissner corpuscles and Pacinian corpuscles do decrease with age, and those that remain are often structurally distorted and less functional. As a result, elderly people are less conscious of something touching or pressuring the skin, which increases the risk of skin injuries. The sense of two-point discrimination decreases, and the elderly have a more difficult time identifying objects by touch. A loss of Pacinian corpuscles also decreases the awareness of limb and joint positions, which can affect balance and coordination. The functions of receptors for proprioception also decline with age, with decreases in information on the position, tension, and length of tendons and muscles. This can further reduce coordination and control of movements, leading to further injury. Among the special senses, elderly people experience only a slight loss in the ability to detect odors. However, their ability to correctly identify specific odors decreases, especially in men over age 70. In general, the sense of taste decreases as people age. The number of sensory receptors decreases and the brain's ability to interpret taste sensations declines. The lenses of the eyes lose flexibility as a person ages because the connective tissue of the lenses becomes more rigid. Consequently, the lens's ability to change shape initially declines and is eventually lost. This condition, called presbyopia, is the most common age-related change in the eyes. In addition, the number of cones decreases, especially in the fovea centralis, resulting in a gradual decline in the visual acuity and color perception of the individual. The most common visual problem in older people requiring medical treatment, such as surgery, is the development of cataracts. Following cataracts in frequency are macular degeneration, glaucoma, and diabetic retinopathy. As people age, the number of hair cells in the cochlea decreases, resulting in age-related sensory neural hearing loss called presbyacusis. The decline doesn't occur equally in both ears. Therefore, because direction is determined by comparing sounds coming into each ear, elderly people may experience a decreased ability to localize the origin of certain sounds, which leads to a general sense of disorientation. In addition, the central nervous system defects in the auditory pathways that can lead to difficulty in understanding sounds when echoes or background noises are present. This deficit makes it difficult for elderly people to understand rapid or broken speech. With age, the number of hair cells in the saccule, utricle, and the ampullae decrease. The number of otoliths also declines. As a result, elderly people experience a decreased sensitivity to gravity, acceleration, and rotation, which may lead to disequilibrium, instability, and vertigo. Some elderly people feel that they can't maintain a posture and are prone to falling. All right, guys, that does it for our chapter on the senses. Thank you very much for listening, and I will see you in the next lecture.